Thank you. So I'd like to introduce Dr. Arpad Danos, who's a scientist here at Washington University and one of the lead editors of the CIViC project. And he's going to give us an update on CIViC and some previews of the CIViC v2 interface.
All right. So yeah, at this presentation, everyone can see the screen here.
I do not see it yet.
Oh, I didn't start sharing yet. That's right. Okay, so here we are. Get these things out of the way. All right, so now people can see the screen.
Yes.
Right.
I'm not sure if you want it to be in presenter mode or this view, but
well, what's preferable? I oftentimes just do this.
it's up to you. Yeah.
Okay, if there's no objections, then I'll just stick with this way. So this presentation is going to be both like a brief. I'm going to try to get quickly through a brief, review of the data model, just so everyone's kind of on the same page, and then sort of a larger exploration of where we're going. And it is associated with the V2. The V2 is a sort of new looking interface for CIViC, which really though has the same functionality. So I think I won't focus on that too much as some of the things that are on our roadmap for the data model upgrades to the way we're thinking about variants in cancer. And so some of it's going to be more, definitely things we're doing. Other stuff is a little more kind of still on the whiteboard planning stage. So you get to see a little bit more of the, it's kind of the work in progress stuff as well. And some of the ideas where they're going. And so some things are more set in stone, others are less. I should try to make that clear during the talk. I think it should be okay. But briefly, CIViC is a project meant to address the bottleneck problem for variant interpretation, which occurs as next generation sequencing techniques have been built into the clinical workflow. Having a large number of variants to interpret can slow things up in clinical practice. So one solution to the bottleneck problem is to employ paid curators and fund the curation activity by keeping knowledge behind a paywall. This sort of stuff, or in private academic silos, restricted access commercial databases. This is one way to approach the bottleneck problem, and I mean it's a legitimate way of doing it. It has its drawbacks. For instance, it's the restricted access being the primary one. Sometimes you'll find with these smaller interpretations also there are things like a lack of provenance, which we tried to be pretty strong about in CIViC. If a variant, if one of our evidence items contains a variant and that's taken from a paper, since we are primarily curating out of PubMed literature, also ASCO abstracts, and we're expanding that to other types of abstracts as possible. We're working with various organizations to do so. But if you see a variant in a CIViC evidence item, you can be, essentially certain that when you go to that paper, you'll find that variant being described in the paper with some sort of what we consider a clinically relevant annotation. And yeah, so that's going to be a pretty strict relation unless editors made a mistake somewhere. So human error might, unless human error out of the picture, then you're going to have a strong provenance for the information we're annotating these variants with. The CIViC principles are like that. It's a free, publicly curated, expert moderated. It's completely free to use with no restrictions, no login required. So public flexible API, and the code is open source. These are some kind of figures that are basically been published recently. Some information on the adoption worldwide. I'm not going to spend a lot of time on this. This talk is really about the data model. But we have a lot of data clients pushing and pulling information. So we're very happy to be part of this sort of larger ecosystem of variant interpretation and clinical cancer sort of reporting and thinking. We also have a strong collaboration with ClinGen Somatic, where ClinGen is utilizing CIViC. We're creating assertions out of submitted evidence. We'll talk a little bit about what those are, and we've also submitted some information to ClinVar, and we'll continue to do so and develop ways of doing so, and also a sort of maybe a closer interaction with the ClinVar data model over the future. But briefly, we public submits curation to CIViC. This will result often in a CIViC evidence item or some other fields in CIViC that are also curated. CIViC is generally hand curated, so we're trying to create a very high level data set that eventually will become large enough to do interesting other things with. Some types of all kinds of data mining can be done, but you'll be dealing with the high value, a lot of intensive effort put into this curation. So I think it's over the years will result in an interesting data set. But generally, the core thing we're talking about CIViC evidence items. These are going to be labeled orange when they're in a submitted state. They're subjected then to curation and moderation. EIDs, evidence can be revised and remoderated. And eventually when an expert curator, and these are a group of curators that are selected to become editor status, go through extra training. So anyone can be a curator, but the editors then are sort of a selected group that have the powers to accept or reject curation. Occasionally, evidence is rejected when it doesn't fit the data model and it can't be moderated. So the fundamental unit of CIViC is the evidence item, and we talked about editors and curators a little bit. Here's an example of an evidence item. There'll be a few more later. This one was submitted by Damien originally in 2018. Predictive evidence type. But we'll look a little bit about that in a few slides. Kind of at the core of CIViC, or historically at the core of CIViC, is what I've been calling, and I've noticed a few other people seem to be leaning in this direction. We're calling it the sort of a somatic, clinically actionable evidence type. Variants are going to be annotated with other types of evidence, but this sort of historic core of evidence items is the predictive therapeutic type, the diagnostic, and the prognostic type. And these together, I'm starting to kind of call the somatic clinically actionable evidence types. And you'll notice that those also overlap well with the AMP type of evidence in the AMP guidelines. We'll look at that in a little bit. There's one other point I'd like to make with this slide. With no supporting clinical evidence, a variant will not appear in CIViC. So that means that, you don't CIViC does not start with a group of variants and then curate them from there, but instead it's when something is taken out of the literature, some clinically meaningful annotation to a variant, it's added to CIViC as an evidence item. If there was nothing for that variant yet, that's when the variant appears in the database, but it's not sort of there in any way a priority. It only appears once something clinically relevant is associated to, and that kind of distinguishes CIViC from other databases. So we have a sort of higher clinical bar for the variants you'll find. So these are then our collection of evidence types that we use. The big three, the predictive, diagnostic, prognostic, also a germline predisposing, and these newer types that we'll be talking about a little bit later. We also have our own evidence levels that we tag evidence items with, and this is a sort of an internal tagging system, an internal guideline only for tagging evidence curated out of literature. So, even if it's like so let's say, just jump here real quick, we're using clinical, case study, and preclinical are the main types of tagging that we tag our evidence items with. And clinical basically be clinical trials, C is case studies, individual or small groups of patients. Preclinical work is tagged D. If the paper has been cited in guidelines or FDA approvals or such, then we'll tag it A. Um, note that like for a very well-established variant like L858R in lung cancer, say its sensitivity to erlotinib, an evidence item describing that relation could still be labeled D if it was an early experiment, say a cell line experiment, because we're curating out of the paper. We're taking these evidence items are chunks of data taken out of a specific paper. So even a highly established relation like the one I just talked about, the L858R, you can still tag evidence supporting that type of annotation with D if you're citing, if you're curating out of a case study, right? Overall field-wide annotation or tagging of these relations will be encountered briefly, and we'll see that we use the amp tiers for that. So here again are I'll give you a few quick examples of these evidence types. So here's a predictive type of evidence. Here we see this is going to be a predictive type of evidence direction supporting the clinical significance of sensitivity response. This is level B because if you take a quick look, this is describing a clinical trial. This is for the variant L858R from the gene EGFR, so it's this canonical type, well-established relation. The drug here is gefitinib, and the disease is non-small cell lung carcinoma. So well-established old sort of relation of targeting one of these primary sensitizing variants, L858R. It's a driver variant for EGFR, and targeted by these first generation gefitinib. By now, of course, there are multiple later generations targeting these. We'll go back here. Note that we can also tag with, we can also annotate with resistance. So, for instance, the famous L858R resistance variant that appears on top of L858R, the secondary resistance variant can be, you can write evidence for that too. I'm not going to show one, but that's we have these annotations are all available. So I'll jump to a diagnostic evidence type. So here, this is a type diagnostic direction supporting a negative diagnosis. So this variant here, FGFR1 fusion, can be used as a negative diagnostic indicator for CML here. This is a level A, okay, because this is actually describing WHO guidelines, which are described in this PubMed publication in Blood. So this describes the guideline, basically stating that it's a negative diagnostic indicator. Moving on, we'll see prognostic direction supports poor outcomes. So this is saying here in a group of, so this is for TP53 mutation. So this variant type is a sort of generalized variant mutation, any type of TP53 mutation. This is level B, so it's a collection of patients. So in this large multivariate analysis, TP3 were correlated with shorter time to first treatment. So it's kind of like a shorter progression-free survival. And I think that's submitted by Alex. And so one thing about prognostic evidence that is a bit of a subtlety and a nuance for the data model I like to point out. How we distinguish prognostic from the predictive therapeutic evidence type, how we think about it. And the way we think about prognostic is that it's going to be a treatment or a drug treatment independent. So if patients with this variant tend to do better, and you're not looking at treatment, you can take a large group of patients treated, given different treatments or no treatment. If you find in general that patients with a certain variant do better or worse, that's prognostic evidence. We can also be a preclinical type of thing. You might show that a variant is associated with something like Ki67. You could then do a level D prognostic evidence. There's a nuance there, but finally, I'll talk a little bit about this categorical, or so colloquially we'll call it the bucket variant. We just saw one, the TP53 mutation. Here's EGFR mutation. And here's specific instances of the EGFR mutation. We can also do C dot level variants, but in cancer, a lot of times our variants are really these sort of P dot level missense mutations. And And so we have a highly flexible variant that allows us to do, this sort of categorical bucket variation, missense variation, or specific versions of missense variation, like pegged with the C dot. So if you go to CIViC, you'll see it's a flexible data model. It's a quick example of how you can get out of like a phase 2 trial, for instance, you could get this sort of EGFR mutation level evidence, but you could also, if say they report individual patient outcomes, you could actually take case study data out of that same phase 2 trial, or out of the same larger clinical trial. So there's a rich type of way you can curate out of papers, and one paper can generate multiple evidence items. So I was going to do a really brief jump over to the CIViC interface real quick. So here's kind of the new V2. Here's a video you can click, and it's a first look at the V2. So if you would be interested to do that, go to civicdb.org and hit this video. So in the quick search, you can put in your favorite gene, EGFR. That didn't quite come up. EGFR. Let's go back and hit this one. And so here's the EGFR gene page with a curator and gene description, some information brought in from the mygene.info. You can load up the collection of variants that are here. We can filter down and find L858. So these are the L858R. So at this point we said L85. So just brought everything to L85. You can narrow it down further if you want, till it's only the things that contain L858R. Then notice how here you already see some flexibility of the variant data model with here it's L858R, and here's a collection, sort of a bucketing of the exon 19 deletion and L858Rs, or the primary types of the main primary sensitizing mutations you see the most frequent. So if we went to L858R variant, you see a curator written description for the variant, information, also coordinates kind of curated, and then a large collection of evidence items derived from different papers, and this is the evidence items that are curated. You could you scroll over and see a brief picture of what the evidence item looks like. You can click the evidence item and see it more up close, what we were just looking at, and you can also browse assertions, which we'll talk about briefly. You can go to genes. You can browse diseases. Here's the different diseases we have, the genes associated with that disease in our database, what's been curated for it so far, and so on and so forth. So I think I'll leave it at that for the database tour. And if you click on this video, so if you click on this video, that brings you to a YouTube channel. And that YouTube channel will also have other videos to check out. So I'd like to talk then briefly about the other main part of our data model, the assertions. So the CIViC assertion, it's a mechanism for summarizing a collection of evidence items for a given variant. So if you have a bunch of evidence items here, and they're all on the same variants and on generally the same evidence type for the same disease, and if it's a predictive evidence sense for the same drug, say, just to be simple, like do an EGFR L858R for non-small cell lung cancer and the first generation tyrosine kinase inhibitor like erlotinib. You have a large collection of evidence from preclinical through clinical trials, and you can collect all that information and write an assertion, and that's a mechanism we have. And the assertions, so I could jump back here. The assertions are here. If you click on one of them, like this is here's one for ALK, maybe go to EGFR. Here's the supporting evidence items for this assertion. Here's a description. This is L858R, so it's a simple one. For the drugs gefitinib, erlotinib, the first generation tyrosine kinase inhibitors. I also have a slide on the same assertion. But that's generally what they're about. But what we can do with assertions, we can start to integrate as well some of the guidelines that are out there, like by now increasingly adopted set of guidelines from 2017, the AMP-ASCO-CAP guidelines for somatic cancer variants, as a way of breaking down variants into the tier 1, 2, 3, 4. We're generally using tier 1 and 2 in our curation and CIViC, although we can do 3, 4. There's a way of looking at evidence. Level A evidence is the FDA approved. Level B is well-powered studies, kind of like the way we're thinking about clinical trials. Level C is going to be off-target use or case studies. Level D is going to be preclinical trials. But what's the difference here is that this is sort of the field-wide way of thinking about it, because what's actually known about the variant. In this case, like if a variant is a level A approved therapy, then you're going to call that a tier 1 level A. And so this is sort of an evaluation, the state of the field evaluation for the variant and disease. I notice how within this paper, they're also breaking it into the therapeutic, diagnostic and prognostic type of way of thinking of variants. So what I'm kind of calling these somatic clinically actionable annotation types. That's just me so far, and again, a few others maybe have said it here and there, but so when we know how there's a way of really fitting this way of thinking about variants into the assertion, because the assertion is a collection of evidence, say from preclinical up through, level A clinical trials. And when you collect that together, you can sort of get a state of the field, and that's where you can add the AMP tier and level. So that's how our assertions work. If I go back to this assertion, you can see that we have our AMP tier and level added to the assertion. It's tier 1 level A in this case. And so again, here's sort of a collect evidence. We can write the predictive, prognostic, or diagnostic assertion, associate AMP tier and level. And here's that same assertion we were just looking at. It's got the variant information, assertion description, assertion type and directions, type predictive, direction supports. It's a clinical significance of sensitivity response. The AMP-ASCO-CAP category is tier 1 level A. And then the supporting evidence items. So currently in CIViC, we have 2 assertion types, but we are going to be expanding that, and I'll talk about that. Then we have this one we just talked about. We also have a germline type of evidence item that I'm not going to, germline type of assertion, which I'm not going to talk about. The predisposing assertion, which allows you to associate a germline variant with pathogenicity from pathogenic down to benign. And this assertion builds in the guidelines from 2015 Richards. And these are codes. You can tag evidence items essentially with codes for those who are familiar with this system. It kind of makes sense. It's it maps onto our assertion model very well, the way that they were thinking about tagging evidence. And that allows you to fold this into a predisposing assertion. But we're going to talk about assertion that follows this same mechanism quite closely. At first, I'm going to talk briefly about the functional and oncogenic evidence types. And these are sort of newer parts of the data model. So I guess this is where the review sort of ends and we start to get into newer territory. So I'm focusing on functional and oncogenic evidence item types. So our main distinction here now, we have 2 types. We've broken this into 2 things, which in the field isn't necessarily always done. And this actually, the main sort of thrust for doing this came from Alex Wagner, who suggested we take what was originally our functional evidence and break it into these 2 types. And after discussing this, we adopted that idea. And this functional evidence type is generally describing data, say from in vitro experiments. They're really looking at how does the variant affect the protein function. So like a basic way to think about it is like an in vitro assay for activity using recombinant protein. That's going to get you functional, CIViC functional evidence, right? Because it's really just how does the variant affect the protein behavior? You're not thinking about it within the cellular context. Whereas the oncogenic evidence, we were thinking about that, and it seemed that, well, I'll get to that in the next slide, I think, but the oncogenic evidence type is supposed to capture clinically relevant information associated with the somatic variance involvement in the tumor pathogenesis. So it's, does this variant affect cell growth? Does this variant affect its ability to metastasize? So like the great way to, an assay that you'd be describing for oncogenic evidence would be, assay for cellular transformation, foci formation, et cetera. So I think I'll just jump to this slide, because the way you really think about the oncogenic, then it actually maps very well onto the notions of the hallmarks of cancer. So you can look through these, the famous hallmarks. Here's the updated ones from 2011, and any of these sorts of processes that are driving cancer, any variant that sort of drives any of these things, you can consider an oncogenic variant. And I'll sort of go back here. Then how are we thinking about functional? Well, functional really does tie closely to the classical Muller's morphs, right? Here's a figure that we haven't published yet. We will publish, but here's sort of the classical Muller's morph for those familiar with it. It's a fairly straightforward scale. You can kind of think about it on this kind of quantitative scale, where a normal variant you associate with 1. That's a variant that's silent, creates unaltered function. As we call gain of function here to be kind of an increased function. As you're increasing a variant function, if the variant increases function, you kind of get a number bigger than 1. You can kind of get a number between 1 and 0, like a reduced function. We call this now loss of function. This is like a reduced function somewhere between 1 down to null, right? Half all the way down to total loss. We sort of annotate that as the loss of function. Then we also add dominant negative. So dominant negative, you got to think of as number between 0 and -1, because say -1 + 1 is 0. A dominant negative will knock out its normal function copy, and then neomorphic is off the scale. So these are the ways we're thinking about functional evidence. So it really breaks right into the Muller's morphs. So it's Muller's morphs on the one hand, and hallmarks of cancer on the other, and that's the way to kind of think about that. So some examples here for again, so here we have autophosphorylation assay. Okay, so that's very much what we call a functional assay. Here we have foci formation assay. So this is really kind of an oncogenic EID, and I don't know, I think my reference is missing here, so I don't remember what paper this came from, but I'll update that next time. And so moving on, we now have we want to think about oncogenicity, because both of these oncogenic and functional evidence types ultimately can be used to start to try to come up with assessments of a variant's oncogenic potential. And so in order to do this in a more formal way, work was done, including by the VICC, the standards for the classification of pathogenicity of somatic variants in cancer. These are oncogenicity joint recommendations from CGC, ClinGen, and VICC. And so I'll quickly, essentially at the core of this is a set of these oncogenicity criteria, which are very similar to the Richards 2015 in terms of the way of thinking about it. But here, these have been obviously thought through and worked out and tested against somatic oncogenicity, somatic variant oncogenicity in cancer. And so you have the strong ones, so these 2 points ones, 1 point ones, and I'm going to come back to this one, the OS2. One thing we want to, that I think wasn't maybe made clear enough in the paper, which we'll get to, is that these points here are flexible. You're not always just stuck with having to use 4 points if you're assessing, say, functional studies to see if there's an oncogenic effect. And notice this use of functional oncogenic is slightly different than CIViC's use. So it's just something to be aware of. That's part of the field. So 2 points, 2 pointer, 1 pointer. These are all generally recommendations, but some are more like set in stone than others, but I think that especially this OS2 is a highly flexible one. I just want to point that out. We also have benign criteria. These are kind of going in the negative direction. So you see that the positive direction, from 0 to 5, your VUS, 6 to 9, you're likely oncogenic, over 10, you're oncogenic. Whereas these negative criteria then push you into the likely benign and benign. So moving from there, if you kind of think about for the second how I showed our predisposing assertion, which didn't go into too carefully, but maybe I should look at one real quick just so we can get a flavor of how that looks. So maybe I'll just go back over to CIViC, browse our assertions, go to VHL, because these are mostly our predisposing assertion types are generally going to be for VHL variants. So notice here how we have our ACMG codes, right, for this germline predisposing variant, and supporting evidence items. And then we have a way of writing these where we talk about which codes are supported and why they're supported, and then also we have a way of linking back to these evidence items using queries, which we still will need to update some of these assertions with. But in large part, hopefully this becomes somewhat clear. This is a predisposing evidence type. Direction supports, in this case, evaluation of pathogenic. So when you sum these up using the Richards 2015 sort of algebra, you'll get a evaluation of pathogenic. I think that's really what you want to look at here: the collection of evidence, the code criteria, and again, this allows us to build a predisposing assertion. So, utilizing these same VICC, CGC, and ClinGen criteria, we're working towards building an oncogenic assertion, which should go live sometime this summer, and that will be exciting to start curating those. And again, I talked briefly about this OS2 curation. So here's like a potential way to do these that I'm proposing, I think, for the FGFR3 curation, and this will be discussed further in ClinGen groups, ClinGen expert panels where this curation is being done actively right now. But I kind of suggest that one would use 2 points for for kind of functional data if you're showing gain of function, because a lot of these assays out there are only going to show like a gain of function type of thing, or what we call in CIViC gain of function, right, meaning increased function. And on the other hand, if for those cases where they really are showing a nice oncogenic effect, either increased growth in cells, or maybe that it's tumorigenic in mice, those are higher level types of preclinical oncogenic data that we can use. We can score the 4 full points. And then in TP53, I did some curation, and I was doing so if an assay demonstrated LOF, I was doing 2 points. If now DN is actually, the dominant negative is actually an important part of P53 function. So I was scoring 3 points to something if it's DN, and DN usually implies LOF. And then if there's like a type of actual oncogenic sort of gold standard assay of increased growth, I was scoring those 4. So that was kind of my thinking there. Now here's something kind of totally different. And this is another brainchild of Wagner, Alex. And so this is a way of, now you'll know how we have these NA, and our clinical significance for predisposing oncogenic is NA. Now, again, our thinking there is because, say we're going with the predisposing evidence, you have a paper, it gives you one code like PM1 or something, and you can't annotate that with pathogenic or likely pathogenic, right? Because to get to those, you need a large collection of codes. So, one paper can't really get you all the way to pathogenic. That's a state of the field evaluation. And so we leave that for our assertions. I think I had a slide with assertions somewhere else, but it doesn't matter. To get to the assertion of pathogenic, you need to sum up a lot of things from a lot of different papers. So that's why evidence items, which again are derived from individual papers, those we just were doing NA. We were not giving them an annotation. But there is still, maybe you want to try to start, annotating these papers with at least the direction, is it supporting something in a direction of pathogenic or oncogenic? Or is it doing something entirely different as well? This notion of protective that was introduced here. So if we look at, say, the second thing here, so this is kind of a scale of therapeutic response. Let's look at this. This is applying to this. So this is the easiest one to think about. So when we think about therapeutic response, we can think about therapeutic sensitivity or therapeutic resistance. And these are kind of like these two, they're actually kind of orthogonal axes, but they're also kind of be thinking about as two different sides of the same thing. But you kind of go from this this clinically significant if it's sensitivity, or it's clinically significant if it's a resistance type of mutation, and then in the center it's not significant because you can have just a passenger mutation. I kind of break this down here in the slide here. So I put this kind of added to this, the notion of primary sensitizing mutation is really on this side. The secondary resistance is being assayed on this side. And here's the slide note. So a predictive evidence can be turned into sensitivity, can be annotated with sensitivity response or resistance, right? And sensitivity response is generally associated with primary sensitizing, resistance with secondary resistance mutations. So, and we have this direction again. We can talk about supports, or we can talk about does not support. And some of our earlier examples showed that, right, for the evidence items, like the I think we didn't have a does not support. We had a negative diagnosis, but we can tag our evidence items with the supports or it does not support. We have this evidence direction, which allows us to assess, mutations for being either primary sensitizing or secondary resistance. For instance, if you encountered a non-small cell lung cancer patient with the novel EGFR mutation, you might ask yourself, well, is this sensitizing or not, right? Is this, and in that case, you would be able to, if the patient responded, say, to administration of a TKI, that'd be a clinical, and you saw no response. Well, that would be sort of a case study level report saying this patient did not respond. They had this rare mutation, they did not respond. So that does not support sensitivity, right? Okay, the details here, if they actually had a primary sensitizing mutation and this was a second mutation, you were querying it, well, is it resistance? Then you might, be thinking about it that way. It's really depending on how you're approaching the patient or the variant, how you're thinking about it. So slightly complex, but I think not too complex, right? So these are the ways we're thinking about therapeutic sensitivity and resistance. We also think about better outcome, worse outcome. So and then so Alex Wagner suggested also adding this notion of pathogenic and protective, since we can have potentially these protective variants, both in a germline and in a somatic context. But this is sort of an active area for debate. I said that some things I'll be talking about are not necessarily, well, they're not set in stone yet. They'll be set in stone once they're in the interface. So again, so this is basically the direction that we're going to be thinking of adding. Instead of having the NAs there, we will have predisposition and protectiveness, oncogenicity and protectiveness as our ways of annotating evidence. Right, and then I think I have an assertion slide way down here somewhere, which I want to go to just to be clear. Or did that not get added? Oh, I think I forgot to add that one. Okay, well, and note though that for assertions, I can just go back up here. Our annotations are set in stone. For the predisposing assertion, we have pathogenic, likely pathogenic, VUS, likely benign, and benign. But for the predisposing evidence items, the units of evidence supporting it, we're going to likely be adding this predisposition, or protectiveness annotation. So this is for evidence. Assertions will still be, as the field likes to think about it, pathogenic, benign, and then evidence will have this, newer kind of interesting way of thinking about things, which we'll introduce to the field. By the way, these things are being discussed also on GitHub. So anyone is welcome to go to GitHub, Griffith Lab, CIViC V2. You can just Google Griffith Lab, CIViC V2, GitHub. I think you'll get, it's easy to find it that way. And you can hear here's the discussion, and anyone in the public can add comments and give us feedback on how we're thinking about this stuff. I'll talk about this next. This is the assertion. But the assertion, I think, is more straightforward. So, right. So I think this just sort of further adds context to the ideas. So again, the predisposing EID, protective, predisposition, the predisposing AID, the assertion, it's just this stuff that's well accepted, well established. And then our proposed oncogenic, our oncogenic EID will have this protective oncogenicity. And then our oncogenic assertion, though, will have this published guideline for assessing oncogenicity, right? But note how for assessing protectiveness, we have no guidelines, no assertion is proposed to discuss this protectiveness. That will purely live in at the level of our evidence items. Whereas assertions, which we consider kind of our higher tier thing, that's the thing that gets pushed to ClinVar. The protectiveness will not be a part of those. Will not be a part of those types of submissions. This will purely live at the evidence level. And here's a touch of thinking. This came out of from Obi on why we might want to also have somatic protective, because somatic protective seems like an odd thing, but one can kind of come up with a mechanism here where that might happen. You could have a somatic. You could have like a bunch of different mutations in a precursor cell, including one of these things that turns out to be somehow protective, beneficial to the patient to have around. It makes the cancer makes it harder for the cancer. You might, do a study and see that patients with this mutation do better. Of course, that also becomes prognostic. So there's still a lot of stuff to think about. But anyway, the model in this slide is talking about how you might have, a bunch of different mutations in the background before you start to then eventually get some somatic oncogenic variants. And then more of them, could push you to transformation, but you still have this around. Now the cancer cells can get rid of this, using loss of heterozygosity, chromosome. Go on.
I'm just curious, based on this picture,
Mm-hmm.
how come what this somatic protective mutation propagated? Because
Because it's just because here, once you have it, you can't just get rid of it. I mean, it's part of the cell.
Why it cannot get rid of it? It's so-called protective, so it does not help cancer. So based on basic evolution, it would disappear.
Well, sure, and that's what I mentioned down here. You'd think that eventually it might get rid of it, but it still has to physically happen. Something has to happen.
But the original question is, how did it propagate originally? How did you pick up evolutionary protective somatic mutation?
It just by luck. I mean, if you're, say, a smoker has a ton of mutations in their
But if
cells,
by luck, you probably it's going to be negatively selected.
right? But you could have luck situations where like the background happens to contain one of these protective ones. The background where these things happen just happens to have this.
Like
It's possible.
It's not. It wouldn't be instant, Dimitri. Of course, eventually, given time, it would be selected away from, but right, there's a time factor where you could have different subclones that have different combinations of background mutations that modulate. So where one variant modulates the behavior of another variant. This is all hypothetical. I don't think there's any, I can't think of any described example in the literature. It's a thought exercise. You could imagine that there's a variant that modulates another variant that causes that lineage to be less oncogenic than some other lineage of cells. And eventually it would lose out to selection, but you could imagine a scenario where there's a time frame in which there's some cells that have these so-called protective variants before the negative selection fully acts on them.
Right. So I think
In evolutionary biology, you can find all kinds of examples of this on the germline side. So logically, there's no reason why it couldn't exist for some time frame in the somatic world as well.
Yes, I think the idea that you mentioned, it's important to keep in mind subclones. For this picture, at least to have a chance of being somewhat realistic, you need to show additional subclones showing it's not rare. It's highly unlikely going to be dominant right away. So I think idea of time is really important, but again, it's tricky. And even on germline sign, you have to keep in mind if what you showed before, just showing the direction, it's gross oversimplification, because protective could be protective in one context, but it could be pathogenic in another context. It could be both. So directionality doesn't quite work there.
The same logic could be applied in a somatic scenario where you could imagine one variant that has a pro-growth advantage in one setting, but then when that subclone metastasizes to another location for some complicated reason, it actually has a quote protective effect or a less oncogenic effect in that context. It's really a thought exercise though, because we were essentially adding this to have like a completeness of a data model, but as of now, we don't have or can't quite imagine an evidence item that would fit here yet. So we shouldn't probably overthink this too much. It's an interesting idea, but it's not going to be a high.
Yeah, I agree. This is really interesting. Thanks. So by protective, could that go, one of 6 ways, like protective in like a therapeutic setting where that second variant would sensitize something to a specific therapy versus not? Or are you just looking at like lessening the effects of the more pathogenic?
I think for the thing you're describing, it's I would go with a different thing where we're gonna, that would be, I would annotate that with sensitizing.
Yeah.
So this is somehow just lessening oncogenicity.
Yeah, I think the concept here is very similar to you have like EGFR variants in a lung tumor, and you have multiple. It's similar, but it's different. You have multiple sensitizing variants in EGFR. Oftentimes, those actually kind of like counteract each other and make your like response less effective to therapy because you have those additional variants, even though if you would see them alone, it's still sensitive to therapy. But then if you see multiple, then it's like less sensitive. So this is it's an interesting concept where it's like lessening the effect of the more pathogenic or oncogenic variant.
Yes, it is similar in that regard, except not, but that's an interesting example you're talking about, which maybe offline would be nice to hear more about those, because those are also something we're interested in curating. Maybe with some of these other changes that we're working towards. So I guess this will just kind of live in a, well, it's under development, but it would live again at the level of evidence items, which is really not the assertion. The assertion is what we sort of present to the world most as our most complete product. This is more theoretical. It's like, well, in physics, sometimes they like symmetry, and they'll oftentimes go with the symmetry in the mathematics, and then eventually discover a particle that actually, works with what they had proposed, what was found in mathematical symmetries. I guess this is a sort of symmetry that has been proposed, and then maybe we'll eventually find some particles or variants that kind of go along these lines. And then, so in our assertion curation data models, if there's some thought, so note then how, again, these are sort of somatic clinically actionable types fit very well with the APASCO CAP 2017 guidelines, predisposing with these ACHG AMP from 2015 and the oncogenic now from the 2022 ClinGen CGC-VIC. So when we choose an assertion, what happens behind the scenes is that an assertion type is chosen, and the guideline associated to the assertion type is sort of hardwired. Well, I think maybe I'll just kind of jump here. So for instance, when you choose this guideline, this pops up, the tier 1 level A, tier 1 level B. And that's going to come up for these types. So when you choose this type, this guideline pops up and these become exposed. When you choose oncogenic, this guideline is going to pop up and these are going to become exposed. But we're also kind of considering swapping, giving more flexibility to guideline choice. So maybe adding this other, maybe when you choose an assertion type, it doesn't just default to this, but it gives you the chance to put in other, and then you might be able to use your own guideline or a different published guideline. And in that case, these will not appear, nothing would appear. And instead you, the curator, would be required to describe the guideline you're using in the assertion description field, that large area for free text. And of course, that would still be just a submitted assertion. Remember how in CIViC expert editors have to come in to approve something for it to sort of have that, for the warning labels to be removed. It didn't mention it, but like any content in the submitted state also has code with warning labels. So, but the motivation for this really is coming from the NTRACs. Now, here's something published, and there's already a well-defined white paper, functional validity assessment. So for NTRAC fusions, there's a SCVCEP, a Semitic Cancer Variant Curation Expert Panel with ClinGen has been working for a while on developing some guidelines for NTRAC fusion oncogenicity. And we want to sort of start to also open up our oncogenic assertion to be able to accommodate these guidelines. So what you would happen under this model is that when you choose oncogenic, you choose a guideline. You either choose the ClinGen CGC-VIC or you choose this NTRAC SCV-CEP. And we might open things up so that users could submit either published guidelines or a well-defined white paper describing the guidelines. And we could add that to CIViC. And then, for instance, these FG things would then appear like as the things. So instead of, you choose this, you get these codes, or you choose this, then you get these codes, right? Which also, yeah.
So one thing, if you go back to the previous slide, I think it all makes sense. One thing to keep in, please go to previous one. It just would be easy for me to explain. So right now, if you think about the germline ACMG guidelines, there is Richards, and then there are VICCs for particular gene. So hopefully the same thing is going to happen now in somatic world. So for example, you mentioned FGFR3, and we'll have hopefully other guidelines like FLT3 and so far, so on. So I think the selection that you mentioned in future, perhaps it would be useful to have something like this one that basically gives the generic guideline, but perhaps below it could be the choice, something like ClinGen CGC, VICC, FGFR VICCEP, or ClinGen CGC, VICC, FLT3 VICCEPs, or something like that.
So you mean like a guideline supporting another guideline? You mean like, because I've thought about that too. I think that's what you're saying, right?
Right. It's
you mean
or just the option to choose the gene specific variant of the guidelines.
Yeah,
I think that
right.
makes sense. Yeah.
Yeah. And so
Once those are published, or I don't know what our criteria should be, but like, I don't see like.
Those would just appear as a separate row. Yeah.
And as you mentioned, it would be very useful because if you do that, in some cases, for example, you will see what will define the criteria differently. Or, for example, we will not use particular criteria at all because for that particular gene, it doesn't make sense. Or we will assign the points different ways. So actually, it would be very, very useful to have this system because it would make it easier for people to curate.
Right.
And ClinGen has this now system for registering these modifications of the guidelines and specifying like
Yes.
the modifications that were made, and so we could refer to that specific registration of those guidelines for each of the VCEPs, and maybe that would allow us to refer to them before they were published, because that can take quite some time. I guess we could talk about the requirements for, do they need to be registered, do they need to be published before we can refer to them, or do we allow like some other internal review to happen, or
So that's
Yeah,
it
so one
opens things up.
possibility would be, because we're talking about VCEPs will go through a ClinGen process, and ClinGen process is documented, and when it goes through all the stages and accepted by the ClinGen, it's published. Now, I'm not really sure it would be published in the system that you're mentioning, but at least it will be published somewhere on the ClinGen site. So
Yeah, that's
I
right.
think they're hoping to support the thematic ones, but I guess we'll see how quickly that.
right, the timing,
Yeah.
So I think there's a few ideas here. Yeah, so we have been thinking about how we would tie that into the ClinGen steps, for instance. But also sort of somewhat, it's perhaps open to the public as well for internal review. They can submit stuff. One important note, I think, at this point, at least, the way that I think I've been thinking about it and proposing in these slides, is that the guidelines does not change assertion clinical significance, right? So you can play around with guidelines, but this remains the same. And if you're thinking about new clinical significances, to me, that means a whole new assertion type. So I'm not sure about time here. There's a few other ideas. Basically, I was going to talk a little bit about ways of combining multiple genes. Maybe I'll just talk about this idea of a molecular profile. We're going to be attaching evidence not to individual variants, gene type variants. Like we're going to start to a newer version of CIViC, which should be hopefully available by the end of the summer, maybe fall. But the idea is that we switch out the current way of doing variants with a molecular profile that allows you to, for instance, take ERBB2 overexpression and PTEN loss. We currently have clever ways of shoehorning this into our current evidence items, which I had examples of in this other slide, but there's not time for that. But we want to start to just come up with, you can do long combinations of variants, put them into a molecular profile, and the evidence is now associated with the molecular profile. And for single variant evidence items like EGFR L858R, that would just be like a molecular profile containing one single variant. And we're also working to expand to this notion of not just genes, but being able to start to try to look at regions. Starting to try to think about maybe regions and associate more complex variant types to that. Kind of an example of that taken from this paper, an example evidence item might look like. So in this paper, just sort of describing 77 Wilms tumor patients tested with these arrays. So this copy number gain here was seen in higher frequency in relapsing cases. So that's like a poor prognosis associated with copy number gain here. So this is a sort of way we would be building. This is a more simple version of a type of evidence item we would be building around regions. I think it's at the hour, so I'll just leave it at that. Unless, yeah, 11 a.m. Okay. And any other questions? Otherwise, thank you very much for your attention.
Thanks, Arpad. That was great. Very comprehensive update on kind of where we're at with CIViC knowledge model. Are there any more questions? I know we're at time. Not, then we'll see you next time. Thanks everyone for joining.
Thank you.